Hello and welcome. Let's start today by talking about the urinary system. Now, when we go through and we talk about the urinary system, here we're going to be able to go through and we're going to be able to see, first of all, the components. Now, when we talk about the components of the urinary system, the components of the urinary system are going to include, first, you can see here, your pair of kidneys, your two kidneys. Next, then we have our ureters, our pair of ureters, urinary bladder next, the urinary bladder we have next, and then finally we have the urethra. We have the urethra. So here when we go through and we talk about urinary system, excuse me, what we'll do is we're going to really tear apart the kidneys, and then we're going to see not much taking place here in the ureters, and then we'll talk a little bit about the bladder, and then again, not much happening with the urethra there either, and we'll go through and get basically these major components taken care of. So first here, let's talk kidneys. When we talk kidneys, the kidneys, their function is going to be, we'll go through and we'll check out. Now here are the major organs we said, we talked about there, right? Kidneys are going to be the excretory organs, are going to be doing a lot of functions. We'll go through all of those in a few seconds. Ureters, responsible for transporting urine. They'll transport this urine from the kidneys down to the bladder, and they'll do that, we'll talk about via peristalsis, we'll see. And then we've got the urinary bladder, and then urinary bladder is going to be storing urine, and then the urethra, which will help to transport it out of the body. So let's go through and let's talk function of the kidneys. When we talk about the function of the kidneys, first function here we can see is going to be that the kidneys are going to be responsible for regulating total water volume, right? We can go through all of these here. I've got my own list. I'll go through there and then we'll hit all of these. Here, I'd like you guys to know when we talk about the kidneys, they're responsible for filtering fluid from the bloodstream. Number one, they're responsible for filtering fluid from the bloodstream. And when they filter fluid from the bloodstream, what they're going to be doing is they're going to be removing toxins, waste, and excess ions, and returning needed substances back to the blood. Also, what can get filtered are going to be certain drugs. So whatever's required then is going to get returned back to the blood. Other functions of the kidneys, okay, other renal functions of the kidneys are going to include now, number one, after filtering fluid then, is going to be regulating the total volume of water. We can see here, so regulating total volume of water, and also the concentration of solutes in that water, and the total concentration of solutes in that water. And we're talking about the total concentration of solutes in the water, we're talking osmolality. Now, we discussed that in the beginning of the semester. Another function is regulating the concentration of various ions in the extracellular fluid. So regulating extracellular fluid ion concentration. Number three, then, we can see then maintaining acid-base balance. Maintaining acid-base balance. Producing hormones. A couple of hormones you're responsible for knowing about will include, here number one, renin. Renin is responsible for regulating blood pressure. Right, you guys know about the Ross system now. I'm sure you guys are experts at that. You're going to need it again in the nursing program. Watch, you do yourselves a huge favor by knowing that when you get there. And your teachers a huge favor as well. And then you can tell them, you came from my class. Now, another hormone we have here is going to be erythropoietin. When we talk erythropoietin is going to be a hormone that's going to be responsible for stimulating red blood cell production, right? Regulates red blood cell production, amongst a couple of other things there as well. Another function we're going to have here of the kidneys is going to be converting vitamin D to its active form. Activation of vitamin D. Right? I told you guys the integumentary system is going to be involved. The liver is going to be involved there. And then you can see here the kidneys will be involved also. Another function here I want you to know is carrying out gluconeogenesis. Carrying out gluconeogenesis, right? Glucose, new generation of new glucose during prolonged fasting. Next, then, let's move down. Let's talk a little bit about the kidneys. So here you can see the kidneys there in detail again. Let's talk location and the external anatomy. Now, when we talk external anatomy and location, the kidneys are bean-shaped organs. They're bean-shaped organs that are going to be found lying in a retroperitoneal position. If you recall from anatomy, right? They're going to be found behind the peritoneum, right? The peritoneum, you weren't able to go into and dissect the kidneys that first day because you had peritonia there. And once you removed that, then you were able to actually see the whole structure of the kidneys. So here when we go through and we talk about then the kidneys, they're bean-shaped organs that are going to be found lying in a retroperitoneal position. In a retroperitoneal position. I thought maybe I had another picture that depicted that's an anatomy. But you have the peritoneal lying basically above. The kidneys are going to be found lying in the superior lumbar region. Found lying in the superior lumbar region. Now the kidneys, they extend from T12 down to about L3. They extend from about T12 down to about L3. Now the right kidney we can see here. The right kidney, I'm sure you learned from anatomy as well, okay? The right kidney, we'll just review this. Not every one of you guys took anatomy last semester, so I like to kind of hit at these main things here. So the right kidney is going to be slightly lower, as we can see. The right kidney is going to be slightly lower because of the spleen, right? Now the pancreas, the uterus, the liver, very good, the liver. All right, we're going to have the liver here, so that kind of pushes down on that kidney a bit. So you can see here it's slightly lower compared to the opposite. So right kidney slightly lower compared to left. Lateral aspect of the kidneys, when you look at the kidneys, the lateral aspect of the kidneys is convex, while the medial aspect is concave. The medial aspect has this indented, indentation, this indented region here that we call the renal hilum. The renal hilum. We saw that with the lungs, the pulmonary hilum. Same thing we can see here. Now, what's happening here? You'll see blood vessels, nerves, and the ureters are going to be entering and exiting the organs. Next in here, when we go through and we talk, let's move here. So we took care of that. Adrenal glands above each kidney, convex, medial, lateral, awesome. All right. So here you can see the kidneys there as well. You got the 12th rib right down here, and you can see that right kidney slightly lower compared to the left. Now, when we talk internal anatomy, that's what we're going to go to now. Here you got all the text and whatnot. Let's look at the picture. So here we can appreciate now the internal aspect. Now, when we talk about the internal aspect of the kidneys, here we can see now, first we have. So now when we check out the internal anatomy, we can see each kidney is going to be divided into, number one, we can see the most outermost region, the cortex. Number two, you've got the medulla, the renal medulla. And then number three, we've got the pelvis, the renal pelvis. So again, each kidney is divided into the cortex, medulla, and the pelvis. Now, when we talk about the cortex is going to be important because we're going to see the majority of our nephrons are going to be found in the cortex. And that's why we'll go through that. Then the pyramids are going to be in the medulla, and the pyramids are going to be important because we're going to see collecting ducts basically coming out from there, and they're going to be opening here at the minor calyces, allowing then urine to make its way into the minor calyx, because before that it's all filtrate, right? Because the collecting ducts are going to be acting on the filtrate and doing whatever they need to do. But once it's removed from basically the collecting duct, then it's going to become urine. And urine is going to go from the minor calyx to the major calyx, from the major calyx then to the pelvis, from the pelvis down into the ureters. So here when we go through and we talk about then the pelvis, so you could see there then as well. Now, we have to make our way all the way to the cortex, basically. How are we going to make our way? We've got the renal artery. So the renal artery is going to take us all the way to the end where the nephron is going to be found, and then we're going to be able to make our way back to basically the renal vein we see here and into the inferior vena cava and back into our circulation. So here then, let's go through and let's talk about the blood supply. Here, you can go through the blood supply, the nerve supply, read that. Let's go through here and let's check it out directly. So when we look at it directly, here you can see from the renal arteries, we're going to be passing through the kidneys, and when we pass through the kidneys, the renal arteries now are going to divide and they'll give rise to then the segmental arteries. The segmental arteries then continue through into the kidneys, and as they make their way through, they're going to go deeper and deeper, and they're going to continue to divide as well. And they're going to give rise to then the interlobar arteries. Here you can appreciate the interlobar arteries. The interlobar arteries then continue deeper into the kidneys, and they continue to branch, and you can see here they're going to give rise to the arcuate arteries. The arcuate arteries. The arcuate arteries then are going to continue even further into the kidneys, and they're going to give rise then to the cortical radiate arteries you see right out here. And you probably saw these inside anatomy as well. They looked very nice. The cow kidneys, they get stained really well there as well. If not, we have them on our anatomy, have them on my anatomy website. Basically the website, if you go under anatomy and you look at specimens gallery, or if you look at the pictures of the specimens, you'll be able to appreciate them there. So here then from the cortical radiate artery then, we're going to reach the afferent arteriole. The afferent arteriole now, we could see when we go through and we talk about the afferent arteriole is going to be now responsible. So then here we can appreciate the afferent arteriole. The afferent arteriole we've seen before as well. We'll check out here when we talk about the juxtaglomerular apparatus, and we go through the nephron, and we'll be able to basically look at all the different components in relation to that afferent arteriole there as well. Now the afferent arteriole then is going to lead us to the glomerulus. The main component, the glomerulus, is going to be the tuft of capillaries, right? That tuft, that bunch of capillaries. So then here what happens at that level of the glomerulus is filtration will occur. Whatever needs to be removed from there is going to be removed. We talked about net filtration pressure before in relation to blood pressure. Well, we're going to talk about it here again towards the ending basically here. And if you didn't get the questions right in the last exam, I mean, I told you in the last exam you're going to get two questions on it. It's going to be a two-part question. And I don't know why you wouldn't make sure you studied that and got it right. I told you that was the only equation I needed you to know there. So here that will be basically appearing again. So if you didn't get net filtration pressure from the last 10 minutes of the cardiovascular lecture before, you can get them basically from here again and get your free points. It's almost a letter grade. So then the glomerulus is the tuft of capillaries. Filtration will occur, and whatever then gets filtered out will get filtered out and make its way into the first component of the nephron, which is going to be the glomerular, basically capsular space. And now whatever doesn't get filtered out is going to be sent back into circulation through the efferent arteriole. The efferent arteriole then is going to give rise to the peritubular capillaries, or the vasa recta, and these are going to be all basically peritubular capillaries. Now blood then is going to be, now here they're really interesting. There's a special set of capillaries here. Now I talked to you about the first function, which is going to be in urine processing, urine formation, filtrate processing. The first function I'm telling you here is going to be filtration, which happened at the glomerulus. Now, second, the peritubular capillaries that we have there are going to allow for basically, you can see there, reabsorption in case we filtered anything out and we want to send it back into the circulation, we can pick it back up. And also, in case we need to remove something that we forgot to take out during filtration, we have the ability to basically do that as well. So that's what these capillaries here are going to be important for. And once then all this is passed, from there then you can see this blood is going to be routed then into the cortical radiate veins you see right out here. From those cortical radiate veins now into then the arcuate veins. From the arcuate veins, the blood is going to make its way then into the interlobar veins. From the interlobar veins, then blood is going to make its way into the renal vein, and then the renal vein, it gets routed, reset into the inferior vena cava. So what we have to look at is all the magic that's going to be right inside of here. All inside of here. Basically what's going to be hooked up to here. So here then we could see, now let's talk about the nephrons. That's what we're going to focus on here. Now when we talk about the nephrons, their nephrons are going to be the structural and the functional units of the kidneys. They're the structural and the functional units of the kidneys. And they're going to be, each kidney is going to contain over 1 million of these tiny blood processing units. Each of the kidneys are going to contain over 1 million of these tiny blood processing units, which are going to carry out the processes that form urine. In addition, there are thousands of collecting ducts, each of which collects fluid from several nephrons and conveys it to the pelvis. And you can see that right inside of here in this picture. So here's one collecting duct, and you can see how it receives many DCTs. I'm sure you learned that in anatomy as well. We talk about this in anatomy too. Now when you go through each segment of the nephron, you can see its histology basically there. We go from having simple squamous cells to podocytes to these basically cuboidal cells to columnar cells. Cuboidal cells you can see, then squamous cells, and then some more cuboidal cells, basically columnar cells, as we pass all the way through the different segments. So here you can see the different segments. You've got, when we go through and we talk, first component we can see here is going to be the renal corpuscle. The renal corpuscle is made up of, you can see the capsule, the glomerular capsule, and then the glomerulus inside. And then here we've got the rest of, basically, you can see the nephron here. You've got the proximal convoluted tubule, the loop of Henle, and you know it's got two segments. You can see here descending limb and an ascending limb. The descending limb is all a thin segment, while the ascending limb is a thin and thick segment. And then the DCT taking us to the collecting duct. So again, we want to pay attention to this nephron, and we're going to see all the events that are going to take place within this nephron. So the nephron made up of two parts. You can see, or the renal corpuscle made up of two parts. You've got here the glomerulus and the glomerular capsule. Next in here, when we go through, let's talk about these nephrons. Each of these nephrons are going to consist of a renal corpuscle and a renal tubule. So each of these nephrons are going to consist of a renal corpuscle, we can see there, and a renal tubule, which we went through and we talked about there, and we divided that tubule up into the different parts as well. Now, each renal corpuscle consists of a tuft of capillaries, you can see right inside of here, that are called the glomerulus, and a cup-shaped structure that completely surrounds the glomerulus, which is called the glomerular capsule. The glomerular capsule. Now, when we talk about the glomerulus, a glomerulus here you can see very nicely. This glomerulus is a specialized structure, specialized for filtration. This circulation, this basically blood supply you see here is very special. This nephron, it's got two basically capillaries on each side of it. So here, one side you see this tuft of capillaries, and the other is going to be the peritubular capillaries surrounding it. So you got to pay attention to these extraordinary things you see here in relation to some of these organs. Now, here again, each renal corpuscle is going to consist of a tuft of capillaries and a glomerulus, right? A tuft of capillaries that we call a glomerulus, and a cup-shaped structure that we call the glomerular capsule, which is going to be found completely surrounding the glomerulus. Now, this capsule is made up of two parts, you can see. First, you have a parietal layer, and then you have a visceral layer. Right? The parietal layer you've seen is basically the outside. The visceral is on the organ itself. So you have the same thing here. Now, here in anatomy, you looked at the simple squamous epithelium in relation to the kidneys, and you had to look at it in relation to the capsule. This is where you saw it. This is what it looks like right out here. And on the inside, what we have then is going to be, you can see here, our blood, basically, filtrate membrane. This is the visceral layer. The visceral layer is very important. You probably learned that in anatomy. And I'll go through it here real quick as well. We're going to go through it in way greater detail as we progress. But here you can see it's made up of a few different parts. First, you have the endothelium, and it's fenestrated endothelium, meaning it's got a whole bunch of openings. Fenestrated endothelium of the glomerulus, right? That's going to be on the inside. And then you can see here a basement membrane. And then here you got your podocyte. Now the podocytes have these little foot-like processes. Those foot-like processes are going to help cover all those fenestrations so that way anything and everything can't just make its way out. Also, the basement membrane has a negative charge to it, which is going to help repel certain substances. So these components here are going to be beneficial. And again, I'm talking about these beneficial components, but when you get to patho, you're going to have to recall all these things can have something wrong with them too. It's like your house, just like your car. You got all these new electronic cars nowadays, and all this fancy schmancy stuff. It's no fun. I don't know how to do that stuff. I think I'm gonna have to learn mechanics and get into that now to learn all that stuff. But not up my alley. I'm not a fan of all that stuff. I like that old school stuff. So here again, every one of these components can have something wrong with it. So that's what you're gonna have to learn when you get to pathology. But again, do yourself a favor and know these parts before you even get there, because when they're going to be talking to you about having immunoglobulin deposition in relation to the podocytes, you don't get yourself all caught up and wondering where is that, what's that, and where is that happening and whatnot. So here then we can see when we talk about the glomerulus, then the glomerulus is specialized for filtration. We said the glomerulus is basically capillaries, and these capillaries are fenestrated, which makes them really porous. And we'll, like I said, we'll go through, we'll check that out. This allows also large amounts of solute, rich, and protein-free fluid to pass from the blood into the glomerular capsule. So here again, you can see all the different parts. Now, when we talk nephrons, I want you to know we have two classes of nephrons. Okay, we have two classes of nephrons. Now here, let's jump forward. Okay, so here we go. We have two classes of nephrons. Okay, we talked about tubular, awesome. Nephrons, we have two classes. You can see here, number one, we have cortical nephrons, and number two, we have juxtamedullary nephrons. The majority of our nephrons are going to be cortical nephrons. Now, here, when we go through and we talk about juxtamedullary, they're going to be found right next to the medulla. Here, you can appreciate basically how those nephrons are going to be found. Here you can see how the majority of the nephron is in the cortex, like I described to you. And here you have the loop of Henle coming down. And here you have the collecting duct inside of here. So here are the two types. Cortical, where the majority of it, even that loop of Henle, you can see is in that medulla still, but you can see the majority is up inside of this cortex. And then here, longer loop of Henle, and you can see it's mostly found within the medulla. Here now, what we can see from this view is showing us, we're going to correlate this view later on as well when we get into talking about the actual functions of the nephrons. So here what this view is showing us basically the osmolarity within that filtrate, within the tubule. So this filtrate that's passing through the nephron, okay, here what happens is as that filtrate is passing through, when it makes its way down into this loop of Henle, here what happens is that filtrate becomes highly concentrated. And here you can see the osmolarity there. 1200 milliosmolars versus 300 milliosmolars when it was more closer to that cortex. Again, why? Because we're going to have water being reabsorbed in this segment of the loop of Henle. So when we start to reabsorb water out of the filtrate, we're concentrating it. And that's why you see it get concentrated. Well, then you're going to say, well, why does it become less concentrated as we move up this way then? Well, to make it less concentrated, what are we going to do? We're going to either add water to it, right? Think about Kool-Aid or something you're drinking. Okay, easiest way to correlate this stuff. Right? How are you gonna now? How are we gonna? We're gonna either add water to it. We're gonna dilute it, add water, right? Or we're going to remove those ions. So here, what happens is PTH, parathyroid hormone. You guys probably remember. Now you have to remember it again. Comes into the picture, and it's going to be responsible for helping to reabsorb certain components from there, helping to restore. You can see that's one of the major components. Again, we're going to go through there and we're going to check everything out in detail. And here you're going to be able to see then helping to restore that osmolarity as basically that filtrate passes through them. So that's what you're seeing here. And then again, we'll actually see it. We're actually going to see it. Here you can appreciate now. Look at here, the afferent arterial. Okay, from the cortical radiate artery, the afferent arterial, the glomerulus, efferent arterial, peritubular capillaries, and the vasa recta. Okay, all peritubular capillaries. Then they're going to allow for reabsorption. If we need to pick something up out of the tubules, we can reabsorb it. That's so cool. If we have something in the blood we want to remove out, we're going to have the ability to remove it if we forgot to filter it here, still as it passes through. It's awesome. Kidneys are awesome. And then here, after this, you can see the peritubular capillaries are going to be hooked up then to, you can see here, the venous drainage. When we talk venous drainage, we've got cortical radiate vein, right? And then making its way basically through arcuate and yada, yada, yada that we talked about. So here then, let's talk about this glomerulus then in greater detail. Let's talk about this glomerulus in greater detail. Now, when we talk about this glomerulus in greater detail, so here when we go through and we talk about the glomerulus okay, is going to be one of the two capillary beds I told you that this basically renal tubule component of the nephron is going to be associated with. Now, juxtamedullary nephrons are going to be associated with the vasa recta, as you've got there. Okay, the same thing, it's a set of peritubular capillaries. Now, when we talk about the glomerulus differs from all other capillary beds because it is both fed and drained by arterioles. The afferent arterial and the efferent arterial. The afferent arterial, I told you before, it arises from the cortical radiate arteries. The blood pressure in the glomerulus is extraordinarily high for a capillary bed. And the reasons why is because, number one, the arterioles are high resistance vessels. And number two, the afferent arterial has a larger diameter than the efferent arterial. There you go. You can see that right here. Look at that afferent versus the efferent. This high blood pressure easily forces fluids and solutes out of the blood and into the glomerular capsule. The efferent arterial drain the glomeruli. And they're going to give rise to the peritubular capillaries, as we saw. The peritubular capillaries, just listening to their name, you can tell they cling closely to the renal tubules. And I showed you they empty into nearby venules. So in summary, the microvasculature of the nephrons consists of two capillary beds. The glomerulus being number 1, which produces the filtrate. And then the peritubular capillaries, or the vasa recta, which are responsible for reclaiming most of that filtrate. Let's talk about the juxtaglomerular complex. The juxtaglomerular complex is a region of each nephron where the most distal portion of the ascending limb of the loop of Henle lies against the afferent arteriole, as we see here in this picture. The juxtaglomerular complex has 3 types of cells. It has 3 types of cells that help regulate the rate of filtrate formation and systemic blood pressure. First, the macula densa cells. The macula densa cells. They are going to be chemoreceptors. They're chemoreceptors that respond to changes in sodium chloride content of the filtrate. Secondly, the granular cells, which are also called the juxtaglomerular cells. These are enlarged smooth muscle cells with prominent secretory granules containing renin. Granular cells act as mechanoreceptors that sense blood pressure in the afferent arteriole. And then third, the extraglomerular mesangial cells. They're responsible for passing regulatory signals between the other 2 cells. And again, you can appreciate the whole juxtaglomerular complex right inside of here. So again, 3 types of cells: macula densa cells, granular cells, and then third, extraglomerular mesangial cells. Let's move to then kidney physiology, and let's talk about the mechanisms of urine formation. Now, when we talk about the mechanisms of urine formation, I'd like you to know urine formation involves 3 major processes. Number 1, you have glomerular filtration, which is going to take place in the renal corpuscle, and it produces a cell and protein-free filtrate. That's very important. Number 2, we have tubular reabsorption. This is a process that reclaims. It reclaims what the body needs. And then third, we have tubular secretion. It's a process that selectively adds to the waste. You can see here 180 liters of fluid are processed daily, and only 1.5 liters are going to be forming urine. So here we saw all 3 of these different processes after they were mentioned here. Okay. So here then, we can see all 3 of those processes, number 1, number 2, and then number 3 occurring. let's go through the first process, glomerular filtration, step 1. Glomerular filtration is a passive process. It's a passive process in which hydrostatic forces, okay, hydrostatic pressure, you can say, hydrostatic pressure forces fluids and solutes through a filtration membrane. Next, we're going to see the glomeruli. I want you to view it as a simple filter, So when we talk about it being viewed as a simple filter, let's look at that filtration membrane then in greater detail from what we saw before. Now, this filtration membrane, it's found lying between the blood and the interior of the glomerular capsule. It's a porous membrane that allows free passage of water and solutes smaller than plasma proteins. The filtration membrane has 3 layers, which are, number 1, the fenestrated endothelium I spoke to you about of the glomerular capillaries. Number two, we're going to see, now coming back to number 1, here's all those fenestrations. Here's all those fenestrations now. You can see in greater detail, the pores, the tiny openings. These fenestrations, these pores, they allow all blood components except blood cells to pass through. Right? We don't have red blood cells and our white blood cells in our urine. Well, there you go. Next, you have the basement membrane. The basement membrane, which lies between the other 2 layers. It forms a physical barrier. You can see in here, it forms a physical barrier. So here we saw those fenestrations on this side, and that endothelium. Next, you have the basement membrane. The basement membrane forms a physical barrier that blocks all but the smallest proteins. Also, so number 2, it has a negative charge, I told you. It has a negative charge which repels plasma proteins. Do we have proteins in our urine? Shouldn't. So you can see red blood cells, white blood cells, right? Proteins stay inside. Supposed to. Third component then are going to be the foot processes of the podocytes. Look at those podocytes in greater detail here. Look at their foot processes. So foot processes of the podocytes of the glomerular capsule. They have filtration slits. They have filtration slits between their foot processes. So if anything makes it through the basement membrane that shouldn't have, it gets stuck here. Molecules that are smaller than 3 nanometers, such as water, glucose, amino acids, and nitrogenous wastes, are able to pass freely from the blood and into the glomerular capsule. Larger molecules are barred. Larger molecules and cells are barred from entering the tubules, just like plasma proteins. Keeping the plasma proteins in the capillaries is very important because it's going to help maintain the colloid osmotic pressure of the glomerular blood. preventing the loss of all its water into the capsular space then. If there are proteins or blood cells in the urine, it can indicate, usually or sometimes, a problem with the filtration membrane. Now, the principles that govern filtration from the glomerulus are the same as those that govern filtration from any capillary bed. Okay? So here you can see that filtration membrane in greater detail. So let's talk outward pressures. The outward pressures promote filtrate formation. First outward pressure that you have to know about is going to be hydrostatic pressure in the glomerular capillaries. Hydrostatic pressure in the glomerular capillaries is going to be abbreviated as HPGC, as we see here. It's the chief force pushing water and solutes out of the blood and across the filtration membrane. The glomerular blood pressure is extraordinarily high. Look, it's about 55 millimeters of mercury. Much higher compared to what we saw in relation to the capillaries, if you remember. Next pressure is the colloid osmotic pressure. The colloid osmotic pressure. Now, when we talk about the colloid osmotic pressure, you'll see the colloid osmotic pressure is going to be a pressure that's going to be abbreviated as OP, capital OP, but a C and an S here. So that's colloid osmotic pressure in the capsular space, because it's going to be a pressure, again, pulling. It would pull filtrate into the tubule. This pressure is 0 because no proteins enter the capsule. Does that make sense? No proteins in here. They shouldn't be filtered out. So there should be none here, so the pressure is going to be 0. If there were some in here, that would be another pressure pulling those components out. And we would have then another arrow, but facing this way. So again, these are outward pressures. Inward pressures then, inward pressures, they inhibit filtrate formation by opposing hydrostatic pressure in the glomerular capillaries. The hydrostatic pressure in the capsular space is the pressure exerted by filtrate in the glomerular capsule. So that pressure, you can see, is about 15 millimeters of mercury. Right? Because whatever's getting filtered out, that's filtrate. Next then, we have the colloid osmotic pressure in the glomerular capillaries. The colloid osmotic pressure in the glomerular capillaries. It's abbreviated as OP, and you can see subscript GC. This is the pressure exerted by the proteins now that are in the blood. If you recall from anatomy, you talked about which protein is going to be found mainly inside of blood. That's albumin. Mainly by albumin. So what we're going to do is we're going to use all these pressures again to calculate the net filtration pressure. So here, when we talk net filtration pressure, let me turn off the subtitles. I thought I knew how to do it really quick and fast. Okay, that's okay. Let's do this. So here we can appreciate the equation. Much easier. Very good. So, yeah, there's a will, there's a way, I guess. I was getting frustrated. So here we can see net filtration pressure. Again now, so what we do is we're going to add, you can see, two forces, right, going the same direction, and then we're going to subtract them from each other, and we're going to get our net filtration pressure. So here you can see we're going to get HPGC, and then if we had our other pressure that's going to be exerted out, which would be OPCS, right, from the proteins out here, that'd be another arrow basically coming this way. We would add those two together. But since we had a zero there, we're not going to even bring that one into the equation. But you got to be able to add that in there. You got to be able to understand that. Just like you're seeing here on this side. HPCS and OPGC. Again, forces going in the same direction. We add them together. Then we're going to get a number here. We're going to get a number here. And then we're going to subtract them from each other. And that's going to tell us net filtration pressure. So here then, you can see HPGC plus it would be, let me write that in. Or let me see the next slide. Okay, so here you can see it would be, right, NFP is going to be equal to HP and then GC plus OP, right, CS from the capsular space. We're going to subtract from, we'll subtract those numbers from, then here we have HPCS, right? So HPCS, this is your equation. Make sure you know this. This is one way to do it. There's many other ways to do it. I don't care how you do it, like I told you the last time, as long as you give me the right answer. I mean, I don't know how much easier I could make it. After that, I guess I'm just giving you the answer. So here's your equation, 100%. You're going to see this on this test too. And then GC. So let's plug and play. So we've got a number here. We don't have one here. So we have that number there. It's 55. Our numbers here equal 15 and 30. So we plug and play. You can see here then 45. So 55 is what we get here. Minus 45 is what we have here. So what are we going to be equaling to then? 10 millimeters of mercury. A movement out of the glomerulus and into the capsular space. Right, the last thing we need is stuff moving back in. Kidneys aren't working then. Could be very dangerous. So next then let's talk about glomerular filtration rate, or GFR. Glomerular filtration rate, or GFR, is the rate at which the kidneys filter blood and form urine. So GFR and form filtrate. So GFR is the rate at which the kidneys filter fluid and form filtrate each minute by the combined activity of all the 2 million plus glomeruli of the kidney. Now, factors affecting filtration rate at the capillary beds are going to include, number one, the total surface area available for filtration. Number two, the filtration membrane permeability. And then number three is net filtration pressure. In adults, the normal GFR in both kidneys is 120 to 125 milliliters a minute. So that is, again, filtration. Everything happening at the level of the glomeruli. Next step in urine formation then is tubular reabsorption. Now, tubular reabsorption reclaims most of the tubule contents and returns them to the blood. Tubular reabsorption is a selective process that begins as soon as the filtrate enters the proximal tubule. To reach the blood, reabsorbed substances follow either the transcellular or paracellular routes. Here we can see them. Depending on the substances transported, the reabsorption process may be passive, where no ATP is required, or active, where ATP is required. So here we can see at the level of the PCT how certain substances are reabsorbed. Which ones involve primary active transport versus secondary active transport versus passive transport. And then you can also appreciate each of those different proteins and channels that we have there as well. So let's go through reabsorption. Let's talk about reabsorption in relation to the PCT. When we talk about the PCT, the proximal convoluted tubule, is considered to be the workhorse of the kidney. It has the ability to reabsorb 65% of the filtrate. It's a site of most reabsorption. All nutrients are reabsorbed here. When we say all nutrients, this includes glucose and amino acids. 65% of sodium and water are reabsorbed here. Many ions. Okay, and here you can see basically in relation to uric acid and urea. Now, when we talk about the PCT, when we talk about sodium ions are going to be reabsorbed, you can see here, thanks to primary active transport via the basolateral sodium-potassium pump that you just saw. Virtually all nutrients, glucose, amino acids, vitamins, some ions, will use secondary active transport with sodium. And then you can see here chloride ions, potassium, magnesium, calcium, and other ions. Passive paracellular diffusion, which is driven by electrochemical gradient. Bicarbonate ions, secondary active transport that's linked to hydrogen ion secretion and sodium reabsorption. Water, osmosis, aquaporins, right? Lipid-soluble substances, passive diffusion. Urea, primarily passive paracellular diffusion. We talk about nephron loop, the descending limb, water, osmosis. I showed you how then the osmolarity there is going to become more concentrated. You can see why water is being reabsorbed. Now in the ascending limb, you can see sodium, chloride, and potassium, thanks to secondary active transport of chloride, sodium, and potassium via sodium, potassium, 2-chloride co-transporters in the thick portion. Calcium and magnesium, passive paracellular diffusion driven by electrochemical gradients. Reabsorption in the DCT in the collecting duct. We can see here sodium and chloride, primary active transport, right? Primary active sodium transport at the basolateral membrane, secondary active transport at the apical membrane via sodium chloride symporters and channels. Aldosterone regulated at the distal portion. Big Al comes into here, so you got to come back and remember all of that. Calcium, passive uptake via PTH modulated channels. The collecting ducts now, sodium, potassium, bicarbonate, and chloride. You can see again, sodium is going to require aldosterone. Water, osmosis. ADH is required here to insert aquaporins. So this is where ADH works at. And then urea, facilitated diffusion. So it's all reabsorption happening in these different segments. Transport mechanism I want you to understand as well. Transport mechanism, there's a transport maximum for nearly every substance that is reabsorbed using proteins in the membrane. TM, you can see abbreviated here. Transport maximum represents the number of transport proteins available to ferry particular substances. Now, I want you to know when transporters are saturated, the excess is going to be excreted in the urine. That's like in hyperglycemia, right? High blood glucose levels. We're going to have high blood glucose levels, and they're going to cause increased glucose to be filtered into the filtrate. But the PCT is going to reabsorb, right? Normally speaking. But if we're in hyperglycemia, the PCT is at transport mechanism. It's at transport maximum. So all the proteins there are busy picking up all that glucose and sending it back into the peritubular capillaries. But there's more glucose than there are proteins. So what's going to happen? Glucose is going to be found in the urine. So it's important you understand reabsorption thoroughly. Especially here with all these hormones coming into play as well. Third, then we have what we call tubular secretion. This is reabsorption in reverse. Selected substances move from the peritubular capillaries. Substances move from the peritubular capillaries through the tubule cells into the filtrate. Urine, eventually excreted, contains both filtered and secreted substances. Almost all of it in the PCT. And here you can see the various substances that can be secreted out. Very important in the disposal of drugs. So here you can see everything basically summarized in relation to reabsorption and secretion. And here you can see we're basically going through and you're able to understand and appreciate how water is being picked up, and here you can see the osmolarity becoming even more and more higher, up to about 1200 milliosmolars as we've described. And then here, sodium chloride being reabsorbed, allowing the osmolality then to go back to about 100, 300 to about 100 milliosmolars. Same thing here. Next thing we have micturition. Micturition, also called urination or voiding, is the act of emptying the urinary bladder. In order for micturition to occur, three things must happen simultaneously. Number one, the detrusor muscle must contract. You have to have contraction of the detrusor muscle thanks to the autonomic nervous system. Number two, the internal urethral sphincter must open. You have to have opening of the internal urethral sphincter, right? Again, autonomic nervous system, because we're talking about smooth muscle. And number three, the external urethral sphincter must open. You have to have opening of the external urethral sphincter by somatic nervous system. Reflexive urination, urination in infants. Distension of the bladder activates stretch receptors. You have an excitation of parasympathetic neurons in the reflex center in the sacral region of the spinal cord, leading eventually to contraction of the detrusor muscle. A contraction, opening of the internal sphincter, and inhibition of the somatic pathways to the external sphincter, allowing its relaxation and opening to occur. Pontine control centers mature between ages 2 and 3. Pontine storage center inhibits micturition. Here, that inhibits parasympathetic pathways, excites sympathetic and somatic efferent pathways. And then here, when we have pontine micturition center basically promoting micturition, here this excites the parasympathetic pathways, this inhibits sympathetic and somatic efferent pathways. And here, then again, you can see basically the same thing as we described. Stretch receptors are going to then involve the brain, let the brain know what's going on, and also the spinal cord. So here you can see urinary bladder fills, stretching the bladder wall. Afferent impulses from the stretch receptors are going to send signals to the brain and also to the spinal cord, increasing parasympathetic activity, decreasing sympathetic activity. And then from the brain also we have the same effect, and decreasing somatic motor nerve activity, helping all to lead to micturition, versus inhibition, where we have a decrease in parasympathetic activity. All right.